All right. So Keoni has decided to subject himself to your heckles and your ridicule. Or praises, right? Praises. Maybe praises. We hope praises. And Keoni probably thinks I don't have anything to say about him, but, you know, we all troll on the DIY bio mailing list. And I kind of wonder if he ever goes to school or anything, because, like, every 5 minutes there's a post by him on there. Right, Keoni? Mom, what do you have to say? What does he do? Does he just sit at home and do science? Post on the DIY bio mailing list? Maybe. All right, but I have no idea what he's going to talk about, but he's going to hopefully blow our minds, right, Keoni? Come on up here. Let us have it.
Hello, everyone. So, my talk today is named "Making Life," and you'll probably see why in a second. So, who am I? I go by the pseudonym name Coing on the DIY bio forums, so people can't Google my name when I answer questions. I'm a high schooler, senior, so I'm going to college next year. Hopefully, I'll get into Stanford. And I'm a DIY biologist, and I've been doing this since about 7th grade, so quite a few years, 4 or 5. So I've built a home lab, kind of like Sebastian's, but a bit scaled down, meant for mainly bacteria work. And through the time I've been working, I've always been inspired by small organisms. So I've built a lab because I wasn't allowed to work with small organisms at the lab I work at UCI. So I built a lab specifically for looking at small organisms and what it means to be alive. So this is Mesoplasm florum. It's related to mycoplasm. It's really small. It's 800 kilobase pairs, which is a fifth the size of E. coli. And that makes it very simple. It's fast growing. It's a nice organism. And so using this organism, I really want to learn, what is life? Minimal genome. So I've been progressing, trying to characterize Mesoplasm florum. What my idea about the organism is that I want to have it open so anyone can use it, instead of just like Craig Venter in his ivory tower with his synthesized genomes. I think that everyone should be able to create life, and everyone should be able to, make an organism specifically for a certain purpose. Craig Venter's organism has taken millions of dollars and lots of years of professors and graduate students to do something that I think can be easily distributed and done by many people. So, of course, first, before I want to delve into how I want to make this a more distributed organism and how we can build it ourselves, my progress on characterizing the organism as a model organism. So I've gotten one strain from Tom Knight, who, when I asked 3 years ago, didn't send it to me until he saw more of my posts on the DIY bio forums and learned I was legit. And I've gotten media, and I've gotten cells growing, so that's a picture I took. It's one of the second pictures online of Mesoplasm florum. They got the genome sequence from the Broad Institute in 2004, after which they pretty much completely abandoned the organism. I'm not sure why. I think they just lost interest, and so no one's really been working on it, except one Canadian lab. And that Canadian lab, I contacted them, and I said, can I please use some of your strains? And they said, sure thing, we'll email you right back. And it's been 2 weeks. They also haven't given me, plasmids. I also contacted RNA sequencing companies, and they haven't really responded to me either because, you Google my name and I'm 17 years old. So, you might know, I'll first explain the analogy I'm going to try and make here. So there's a difference between different genome engineering approaches in order to understand what life is fundamentally. There's the pocket of people who want to make a minimal genome, the smallest it can possibly be. And while that's useful, we don't actually get to know a lot about the organism because we're just deleting things. However, if we go in and try and build an organism from the bottom up, semi-bottom up, you can learn a lot more about its biology because you have to optimize certain parameters in order to learn the interactions between separate genes. So, here I'm going to make an analogy between Minix and Linux. Most of you know Linux. Actually, probably all of you know Linux. But how many of you know what Minix is? So, very few of you. Minix was an operating system created for educational purposes. And Linux was actually based off of Minix. Yet none of you have ever heard of it. That's because Minix was only used for education, and the creator decided to never release it to the public to allow other people to work on it, unlike Linux. And so you've really seen where that approach got them, not very far. Just like what Craig Venture is trying to do with his organism, that he will not pretty much share with anyone. I've asked him for even just plasmids to help me engineer mine, and they said, I'll get back to you. So, the minimal genome. What advantages it has? Well, first of all, it's specialized, it's slow, and expensive. Because when you minimalize a genome, you're just concerned with taking out genes. And when you're just concerned with taking out genes, their approach was very synthesis-oriented, which I'll get to why that is an inferior approach as well. Not inferior, it's just different. It's expensive and slow because you have to synthesize, then you have to clone, then you have to put it into an organism, and that takes a long time. There's no distribution for Craig Venture's approach because, he lives in an ivory tower, and you can only do minor rearrangements of the genome because you have to synthesize it every single time. So what would a modular genome be? So this was an idea I've been dwelling upon for a couple of years that I'd really like to pursue, and I'm currently pursuing, to make a standard genome for Mesoplasma florum. So a modular genome, what I envision would be standard. Anyone could work with it. All the protocols would be laid out in a simple, easy to follow fashion for anyone to be able to do it. You can enable cheap testing because you can just conjugate this entire construct in instead of having to go through yeast tests, which, if anyone knows how to agarose plug bacterial artificial chromosomes, they would know it's very difficult. And when we modularize something, we put it into different parts that can be replaced. We can have easy distribution networks and contribution networks in which people can work on certain parts of the project and certain areas of the project while not having to be part of a whole. And by enabling this, you can have large rearrangements of the different gene clusters, enabling you to genetically modify an organism with much more power than you could ever do before. So synthesis. A lot of people are like, oh, hey, synthesis costs are going down. We should just synthesize organisms instead of, assembling them in some different way. But I draw the analogy here to compiling versus a binary package. Now, most people have never installed a software on their computer that they had to compile themselves. That takes a lot of time, a lot of processing power, and a lot of energy, just like synthesis does. And why would we synthesize something if we can get it just, mailed to us much quicker, much easier? So that would be a binary package, a DNA that's already assembled that we can just use. And so many people would draw a line to, the BioBricks. And while BioBricks has been useful in the past, I don't think it's the future. So this is called Linus's Law, The Cathedral and the Bazaar. It's a great book. But given enough eyeballs, all bugs are shallow. So what this means is if we get our organism out to enough developers, enough in the open source community, then the bugs and some of the things that go wrong with our organism will eventually be, we can find solutions to them much easier because we have a lot larger diversity in our community rather than, Craig Venter, who just has all his buddies in his little lab. So I would imagine that in order for us to distribute it, we need a new distribution method because while BioBricks are great, they're mostly just used in iGEM. And iGEM, they say they're open source, and I guess they are, but they charge $3,500 to, compete in their competition. And that's about how much my lab is worth. So I can't pretty much afford to ever get, the full distribution of iGEM parts. It's actually cheaper for me to make my own little distribution. And so I don't think that BioBricks are the way to go because they're too slow. So instead I would propose a Golden Gate method, which Golden Gate is just modular. You can put together 10 parts at once. So 10 parts. What would I need to construct an organism with that? Say if we have all the genes that we have for mesoplasm form, about 700, 800 of them, we put them into plasmids. That's smaller than the iGEM, what they send for iGEM. We could actually construct full operating organisms from scratch using a small distribution kit, the size smaller than the current iGEM kit. So I've been developing technologies in order so I can distribute plasmids easily to people. The iGEM method is to distribute plasmids in just dried plasmid stalks and plates. That's good if you have all the resources to do that. Now, most people don't. So I've been working on some plasmids. I've gone through 3 iterations so far. This is on Golden Gate. You can put together 10 parts about maximum for general use. So what I'm really concerned about is, like, how do we get it out to everybody? If we want to be able to, going back, make all the bugs shallow and all of my organism very well characterized, I have to get out to the greatest amount of people I can. So how do I do that? So what I envision is phage distribution, in which we package all the parts of the genome into phage particles, which can be made essentially for free. And I'm in the preliminary stage of testing this. And with plasmid stocks, you have to mini prep. You have to then store it correctly. You then have to send it to the people, and they have to retransform it and verify. If I package it into phage, I can get the cost down to 0 for packaging, get enough in a single reaction to send to every lab in the US. In fact, a liter would be able to go to every lab in the world for about the cost of 1 maxi-prep. And if you want to retrieve it, you just mix it with some bacteria, and you're good to go. And so if you can distribute all the plasmids that the DIY bio community produces in this format, it'd be extremely easy and cheap to distribute and contribute plasmids to a central repository or source, a method that would be much cheaper than the iGEM one. And so then what else can my distribution system do? Well, I would like to include a conjugation system, meaning that if you have plasmids that you made for this organism, you can put it into any other different organism. So in the process of trying to get distribution down to make my minimal and modular organism more accessible to everyone, I investigated general strategies to make it more worthwhile for you, the people of the DIY community, to adopt such a method. This one would be a conjugation method. Now, what I'm looking to do is have a single organism that you can have, and you put a plasmid in it. Just infect it with a phage particle. Pretty much it's just mix and go. And if you put it into this organism, you can directly mix it with any other organism, such as plants, yeast, other bacteria, even the bacteria I want to modularize. And it will immediately transform it. So that takes down weeks, sometimes it would take to characterize an organism, make comp cells for it. All you have to do is get my strain, get the virus, infect, and then put it into your new strain. And I think that would have a lot of advantages versus the current method of people just sort of distributing plasmids and saying, have fun transforming that. And so finally, DNA assembly. So I say it's an efficient crossroad because the crossroad between DNA assembly, which is what I'm looking at, and what I want to make easier for everybody, has a direct correlation to how many genomes we can produce. In the future, I believe that genomes will be more understood and we can produce them much easier, which will allow rapid advances to biotechnology. And in order to be able to produce more genomes and more variants of genomes, you have to have an efficient way to make them. Some people would say synthesis, but that's like compiling. It will take a long time and it will still be costly. Always cost more to synthesize something from scratch than get it from a cell. So if I can learn genome production, that pretty much solves, well, I'll have to learn DNA assembly to get genome production. So the current on the left-hand picture is how Venter currently produces his organism, his minimal organism, through synthesis and then constructing it in yeast and going through many Gibson runs. Now, any of you who have worked with Gibson will know that you have to have new PCR primers for every single single thing that you want to Gibson. That means if you wanted to reproduce his results, you would have to do it pretty much completely by scratch, ordering all your own primers, all your own DNA, and that's if you would even send it to you. Meanwhile, if we put this into, say, a Golden Gate format in which we can allow people to quickly produce new DNA molecules, anyone could do this for very cheap. I calculated it out, and it would take about 4 or 5 reactions in order to produce a full-sized organism. Well, 4 or 5 reactions over time. It would take around 40 reactions or more to get the full product. That's much simpler than what he's doing right here. We can skip the middleman step of synthesis and immediately get an organism that would be useful to anyone to engineer and learn fundamentally what is life. That's really what I want to look at, because when we say what is life, we don't really know what it is. It's fundamentals. We don't know the minimal gene set. We don't know how those minimal genes work with each other. But if we can build a genome to do stuff, for example, we rearrange genes, or we make it do a certain a specific process, or we reorganize these genes. While we are reorganizing, and while we're bug testing in a distributed fashion, we would give information to fundamentally what is life. And so what my goals are is to distribute a large-scale method to give people DNA for cheap, to standardize protocols, which I'm currently working on. I believe personally that anyone should be able to take a protocol from, say, Open Wetware and use it in a home lab. But I found that most of the protocols that I see are specialized towards labs. I think that there should be more open communication with protocols. Kind of going after what Sebastian said on simplifying media, I believe that if we can simplify media for a minimal organism, or for an organism anyone can use, that would advance our knowledge of what is life. And then finally, unlike what I've found in, say, like iGEM competitions or different biology competitions that I've done, I would like to allow anyone to contribute to this project. Anyone that wants to can jump aboard and just help create an organism that would be distributed and be owned by the community, not owned by some corporation or Craig Venter. Thank you. So, right here I'm coingb.io is my main website where I occasionally post on my blog, and then I have 2 other domain names that I'm currently working on, wikis. Any questions?
So first off, I'd like to thank you for shit-talking Craig Venter that much. That was awesome. Second, so do you see what you're doing right now leading to more what happened with Linux, where there's a bunch of different distros like Ubuntu, which like anybody can download and use, and then Arch? Or do you see everybody really like making their own genomes customized for their work specifically? Like how much detail do you think an individual is going to put into a genome?
I definitely see it as a more Linux-based approach. I think that fundamentally that approach in an open source community to build something large works the best. And if everyone, is just contributing their own little things and growing, a community, eventually there will be personality arguments and people will fork off of a project. But that's okay in our case. Once we have the fundamentals down, anyone who wants to fork can, and that would just help the community. Any other questions?
Hi, thank you very much. I was just wondering what your gut is telling you about a potential minimal gene set, whether you think there is one minimal gene set?
Oh, no, definitely. I think that there are many different minimal gene sets that you can have. Like, for example, 2 genes could do the biosynthetic pathway of maybe 3 genes or so. And there are probably a lot of different combinations of genes that you can have that would either be, lethal or allow you to live with extra genes. It would be very difficult to define a minimal genome until we know how every gene interacts with each other. And until we're able to efficiently reorganize the genes, I think that would be a very difficult answer to directly answer.
Why would discovering the the minimum gene set actually define life, though?
I think that discovery of a minimal gene set would be interesting. Just to know what is life, but that's a poor economic argument, of course. What a more economic argument would be is that if you were able to define a minimal gene set and a minimal organism, you would be able to reorganize its genome to be able to do new and novel processes. For example, this Mesoplasm florum, I did a search of its tRNAs and found out that it doesn't have any tRNA for the codon CGG. Now, if you wanted to, say, produce a protein with an unnatural amino acid on a large scale, you could easily do it immediately in this organism, something that's taken E. coli labs years to get down to be more and more efficient. And if we have a small genome set that we know works and that we know how it interacts with each other, you could begin reducing codons, adding more natural amino acids, which would allow you to produce more different medicines and different products of the ribosome in a selectable, biological, in vivo way, which allow you to, more democratize the whole scientific institute. Anything else?
Thank you. Fucking biohackers, they're the shit.